right here. So in the original and in the revote, nobody picked the RGD peptide stick to bind to platelets and prevent sticking. RGD bind to integrin on the endothelial cells to prevent adhering to the veins. I'm not even quite sure what I was making stuff up. Okay, but I have to come up with 4 things. So it really boiled down to these two. RGD peptides bind to the integrin and compete with fibrinogen binding, or RGD peptides bind to the fibrinogen and block its ability to bind to integrin. And to distinguish between these two, it's important, so the vast majority picked that one on the revote. This was a distractor. What's important to realize is who has the RGD sequence? Where's the RGD sequence in this? The fibrinogen. So an RGD peptide would not bind to fibrinogen. Rather, the binding site for RGD is on the integrin. And that's why integrins will bind to fibronectin, because fibronectin has an RGD site. That's why integrins will bind to collagen, because collagen has an RGD site, and they'll bind laminin as well as fibrinogen. So how does that result in blocking platelet formation? And it's sort of illustrated here in a not great way. So this is the site of injury right here. In blue are the RGD peptides. Because it's a peptide, peptides are tiny. They can you can infuse a site, you can make a solution that has very high molar concentration, lots of RGD copies. And so each of because integrin has an RGD binding site, each of these will bind the RGD peptide. So what I don't like about this drawing is it doesn't show that, it looks like the fibrinogen disappeared, but fibrinogen is still there. It's just as able to bind to integrin, but rather it is outcompeted by the vast excess of RGD peptides. That's why the infusion was of peptides, in order to bring up the small so you can bring up a large number. If this molecule, if this integrin was to bind fibrinogen, it's really unlikely that another platelet would also bind fibrinogen. You've disrupted the networking ability of the integrins to cross-link with each other, because each of these is saturated by an RGD peptide. Does that make sense? This turned out not to be, I don't know the reason it wasn't feasible, but it's not how, this didn't work for the company. So it was either making the RGD peptides in a way that was getting them to the site or something. Question here. Oh, so the question was, does aspirin work the same way? So many of you may know that people take low-dose aspirin in order to block clot formation, and it's actually recommended right after a person has a heart attack. That's the first thing you can do, is take some aspirin. And it doesn't work this way, but now I'm curious to find out how aspirin works. I got the same question in the morning class of a person who worked with an EMT, and they carry aspirin with them to give to cardiac patients while they're shunning them to the hospital. There was another question somewhere.
I was going to ask if this is how it's actually done, or if there's some other.
Oh yeah, no, the idea didn't work out. It was it was just in the research phase. So in making a biological therapeutic like this, there's problems with packaging, there's problems with delivering it to the site. There's problems with maybe the RGD peptide degraded, when it hit all the proteases in the cell. How do you encapsulate it so it's available to the integrins, but not to the proteases that are in the cell? So in the extracellular matrix, in the serum. So I can imagine that there were multiple stages at which this might have failed. I don't know where it did. Other questions? Okay, we're done. And this is the last, this class will be on the next exam. But the next class, which is, will start cell cycle, will not be on.